Hello, my name is Jesse, and today I'm going to be sharing a presentation that I gave recently at the ACS conference that happened back in August. And this is about consolidating drug development data within a pharmaceutical laboratory. So to begin with, I want to share about the state of data in pharmaceutical development. I'm sure that there are some people who are in the audience who are familiar with pharmaceutical development, but not necessarily all of them. And sort of what it looks like is something like the following, where there are a lot of groups who are working together in the research process, and they're sharing information through a variety of different mechanisms, including emails. There'll be central repositories. There'll be different formats that everybody is using based on the particular instruments that they have access to and the equipment that they prefer. And you also have like analytical chemistry and process chemistry are a little bit separated from one another. You have contract organizations that they may be involved, often are involved in some capacity, and they may be sharing information once again through emails or through PDFs, dropping it off in central repositories as well that other folks have access to. And ultimately, you have regulatory CMC who are trying to then pull information out of all of this mixture in order to write up regulatory documents that are going to be going to the FDA or other medical, like review agencies. And overall, this leads to a landscape that is very difficult to navigate, and where if somebody needs a particular result, especially if it's something that they haven't generated themselves, it's very difficult to know even where to begin to look. And that's a very real challenge that can really slow the efficiency of the scientists who are working in this field. Now, that is a qualitative view of what we've heard from the folks that we work with in this field, but let's look at a little bit more quantitative view. We did a survey last year, and we found that we were asking analytical scientists how they are managing their analytical data, and over 2/3 said that they find it difficult to access and share their analytical results. So that's an overwhelming percentage, in the sense of it's definitely the vast majority there. But they need this analytical data to do a variety of everyday activities, such as comparing new results to old results, which is a very helpful thing to do to make sure that your results that you're getting today are what you expect them to be. You're providing information to regulatory bodies as well, so that's 71%. So a lot of these everyday kind of activities. Now, one that's worth particularly pointing out is replacing lost/misplaced data. Now, it's the smallest percentage of what's here, but it's particularly important to remember because it's most costly as a condition, as a situation, because it requires time and resources of equipment and people in order to actually replace that data that's missing, and so that's a very real concern. So while it might not be happening every day, hopefully isn't happening every day, when it does happen, it's actually a very real cost to the companies and to the scientists. Now, what's driving this? Why is there so much challenge in finding the results that we need? Data diversity is one of the biggest causes here. So over 90% of the scientists that we surveyed said that they use 2 or more techniques, use 2 or more instruments for analysis, or use 2 or more software applications to process their data. So that is very common. I think that's common for almost everybody who works in pharmaceutical development, where they're going to be using multiple types of data in order to verify their results. So this is the sort of concern. This is the sort of issue that is not a quote unquote problem that needs to be solved. It's not like we need to be doing less checking of our work by comparing the NMR and the mass spec, or the NMR and the chromatography. Rather, we need to think about solutions that allow to use these different kinds of data more holistically and with fewer barriers between them. Now, where is this data then being stored is another question. It tends to be very scattered. It's across multiple different systems. You have Microsoft applications, vendor software, ELNs, LIMS, etc. But one thing to particularly point out here is that the numbers, if you add them all up, get up over 200%. So that means that the average scientist is storing their data in 2 or more places. So that's quite a substantial number, and it means that it's a real challenge to track down and remember where you left your data that you want, or find somebody else's data, because it could be in multiple locations. So for the rest of the presentation, we're going to be drilling into one of those particularly and the challenges that are associated with it. And that has to do with the Microsoft application Excel. This is really important. It's very hard to overstate the importance of this in pharmaceutical development. For a lot of organizations, they really rely on this in order to unify and bring all of their data together, consolidate into one place in order to do their final analysis and compare different routes, or to prepare the results that ultimately go into the regulatory documents that I was discussing earlier. And there's a lot of reasons why we default to Excel. You can kind of export anything into a CSV file or to Excel file. It's very flexible, and everybody has access to it almost, and everybody's been trained to use it as well. But it really doesn't meet the needs of pharmaceutical development scientists, though. This is an example of a template that is used by some customers of ours, AbbVie, and this is what they were using before, and they said that they had to summarize 36 individual spreadsheets in order to fill in this particular spreadsheet that they would use for this part of their process. That is a lot of work to do. There's a lot of busy work and a lot of opportunities for error and a lot of basically babysitting that's necessary in order to have this function properly. So this is a very real challenge that if you're relying on this type of technology to hold all of your pharmaceutical data, it's really just not designed to keep up with this. So the paper that I'm sort of presenting on the results of here was one that was written by some colleagues of mine a couple of years back, consolidating and managing data for drug development within a pharmaceutical laboratory, and is really trying to get at why Excel does not meet the needs of pharmaceutical scientists. And there's a few reasons that they point out, but there's a few here that I'll list specifically. One has to do with chemical structure awareness. So Excel does not understand chemical structures. It does not. You can't give it a chemical structure and it'll figure out what either the chemical formula is, the chemical weight. It's not going to be able to assess how similar different structures are compared to one another, and that means that things like structure-based searching are impossible, or name-based searching can be really difficult too, unless you've inserted that deliberately in the metadata. Like, there's some workarounds here, of course, I understand that some companies are using, but by and large, it's very difficult to do that kind of searching, which is very difficult given the fact that all of this work, it tends to be built around chemical structures. Consolidating the data is also challenging. So you can of course consolidate basically anything that you want into an Excel spreadsheet, but it's a question of how much workup and how much effort is necessary in order to clean up the data to get it into that shared data format. And it's often very difficult and can require a significant attention from some scientists who would rather be doing something else with their time. The data is also dead analytical data. Dead analytical data is what we call it, and that is the idea that you don't have access to the raw analytical information or the process analytical information. You're just kind of given this list of values more often than not, and you can't interrogate this in any way. So if you have something like a chromatogram that you want to recheck to see whether there was a hump on the side of it or whatever, you just have to sort of accept that that's it is what it is of the numbers that you've been given, or you have to then go hunt down the original analytical file, which can also be very difficult to do. Time intensive is another issue that they identified in this for Excel. It just requires a lot of time to do things like clone or template different spreadsheets because there's just not very good tools for that. If you're doing this improperly, you can very easily break things. So it's very time intensive to just manage these things and to babysit them properly. And then collaboration and versioning is another major problem. So that when you have collaboration across different groups who are all sharing these same spreadsheets, if you have one person who saves at the wrong time or in the wrong way or overwrites the data that somebody else has included, there are a lot of opportunities for mistakes to happen there, very small mistakes to cause a lot of trouble. So that once again requires a lot of this babysitting in order to make sure that it functions properly. So, how do we solve for these problems? What's the alternative here? A lot of ACDLabs' software is based on the Spectra Platform, which is a very alternative, different view of how to manage scientific information. We are bringing together multi-technique data, so you can bring together your NMR, your mass spec, your lab reactor data, and read that in the Spectra platform. Is also multi-vendor as well, so vendor agnostic. So you can import data from basically any set of instruments that you want in order to bring them together, compare it, and see the results, and you're not forced to use all your equipment from the same vendor, for example. But it also, on top of this, doesn't have some of the same issues that we sort of identified in Excel. It is a chemically intelligent system, so you can do things like structure-based searching. The analytical data is live, so that allows you to get your hands on that analytical data more easily. And it's built on the FAIR data principles, so findability, accessibility, interoperability, and reusability. Luminata is the particular piece of software that I'm going to be talking about here, and it applies the logic and the power of the Spectra platform, but in the process map as the kind of the unit of analysis. And this allows for some really powerful things that you can do, and I'll show you that in a little bit. That analytical data is then mapped onto individual stages, individual structures within this system, and that will really allow for us to have much better understanding of what's going on in our reaction as we're managing our process and trying to understand our process and looking for things like impurities there. So the study compared the synthesis of agomelatine, so using the data from the synthesis of agomelatine in these two different systems, so Excel and Luminata. The operations that were examined was structure elucidation, process control, and calculating carryover of it. Using Excel, we found that each stage needed to be built separately, so that was very time intensive in order to do that, and that you couldn't just template things and clone things easily. You really had to build these things up from scratch. Consolidating analytical data is very challenging, so that that workup that's necessary in order to get that data into a format that is useful for this ingestion into this spreadsheet took significant time as well. And then it's also just time consuming and error prone to manage this whole system. So it's a very clunky system to use. Now, elucidating structure of impurities with Luminata, there's a lot of advantages here. So we have the process map that's on the top left that we can see where each stage of the reaction is compared to one another and the different compound structures that are within that. We have batch results, so we have our actual analytical data that is available in the software right available there. The data is also available in tabular format, so you can see it in a numerical setting, so in some ways similar to what you would expect in that Excel sheet there. And then you also have reference data as well, so if you're doing comparisons between this stage and that stage, or this run and the previous run, you can make sure that your results are looking like what you would expect they would. When we're doing things such as the origin and fate of compounds, you can see the original compound here, and then as we go through the set of stages, so this is the starting material, the concentration decreases. So it starts at very high, of course, but then it goes down to 0 by the time that we reach stage 5. So that allows you to have that confidence of saying, hey, we've eliminated all of this. It's all been either converted to something else, or it's been purified out. So you have that confidence to know that you've done your controls properly. You can also do things such as calculate carryover. So this is a useful number that it can help you understand how effective each of your steps are at removing the impurities that you're interested in. You can also convert this into a purge number as well, if that's how you prefer to visualize the information. So that is very handy to have access to as well. So how does Luminata then compare to other solutions? So overall, we think that it has a lot of advantages over Excel specifically. It's really been designed to meet these needs of the pharmaceutical development scientist. There are a number of other applications, such as ELNs, CDSs, and LIMS that scientists use as well that do meet some of these requirements to varying degrees. It's going to depend a lot on the particular implementation there, but I think that Luminata particularly has been shown to be really effective for the needs of those pharmaceutical development scientists. So that'll be it with this. Thank you very much for your attention, and thank you to the folks who helped to make this possible. This is the publication that you can see here. If you want to look it up, there's also be a link that'll be in the description for this, as well as a link to Luminata, and you can see that right here, or you can go to acdlabs.com/Luminata to learn more. Thank you so much for your attention once again. Take care, folks.